mechanisms of complement activation in thrombotic microangiopathy. We just heard, and you'll hear it. You'll read it in the literature. You'll hear it in presentations that atypical HUS, CM-TMA is an alternative pathway disease. I would prefer to rephrase that, and really to get you to understand this, I need to do a little bit more investigation into that diagram we saw there. I'd prefer to rephrase it as a disorder of the amplification loop of complement, if you're going to use that term, and you still probably need a stimulus to kick off, right, the amplification loop of complement. So that would be my goal of today's talk. I know, I don't know all of you guys' backgrounds, but to get you to understand the role of the immune system, I do have to dig a little deeper into how those pathways are kicked off, how they're amplified, and then the lab work that I'm doing to try to figure out which way it's going. Is it the kickoff, or is it the dysregulation that's ultimately leading to the damage, the cell damage that you're seeing in complement-mediated thrombotic microangiopathy? All right. We'll try that. Try keyboard. No. So I'm gonna start with a case presentation. We're gonna focus on how the alternative pathway is activated, and then we're gonna discuss how we detect membrane complement activation in comparison to the fluid phase markers. You just heard a lot about some of the fluid phase markers that can be sent very easily. The membrane tests that we do, they are now available, but they're still niche, right? So you have to seek them out, and they're still somewhat expensive. So we're gonna discuss that. And then we're going to discuss what the evidence is that I've done in the lab at Johns Hopkins. And we're going to go through some overview of the prior literature that maybe the classical pathway may be an initiating stimulus leading to the complement dysregulation. I do have these disclosures. That is the assay that I'm discussing and I've developed with Dr. Brodsky at Johns Hopkins is licensed to Makion, and I am entitled to royalties, and I own stocks. So the case presentation is a 25-year-old female with no significant past medical history. She developed fatigue and malaise for a few days. She presents to the hospital. She's found to have influenza B testing, and she's found to have all those markers that we just heard in the last presentation, right? She has thrombocytopenia, she has low hemoglobin, she has an elevated LDH, and her smear has the little triangles on over there that show that she has a thrombotic microangiopathy. And they do send genetic testing at the time, and it shows that she has a pathogenic CD46 variant known to decrease expression of CD46, and her father does too, but her father was never affected by that CD46 variant, and he's in his 50s and 60s. Why did he never get the disease, but she did? Was it bad luck, or what was the trigger for the disease? So this does bring up those mysteries and things that we still don't really understand that well about thrombotic microangiopathies. That is, only 50 to 60% of patients will have a genetic variant that we can detect on genetic testing. So it's not diagnostic. It doesn't tell us you didn't have a thrombotic complement-mediated disorder. And on top of that, you don't need even if you have a variant, that is, the father never got the disease. So if you look at family members with known pathogenic variants, the best evidence we have is the penetrance is only 20%. Over your lifetime, if you have a pathogenic variant, your chance of getting disorders is only 20%, right? So that's not saying that it is everybody with that variant is going to get it, and it's not showing you that the amplification loop is the primary reason for getting the disorder. Secondarily, it's been demonstrated quite a few times that if you look at CD46 in particular, although a lot of the variants, the factor H, the factor I, those are fluid phase regulators of the amplification loop. CD46 is on the cell membrane. And if that is the membrane of that person's cells in the kidney and their endothelium. If I take the blood out of that person and I separate out all the cells and I test their serum, I can see the complement dysregulation. But the CD46 is not in their serum. It's not in their blood. That shows you that there is something else going on in the serum and the blood other than that CD46 mutation leading to the disorder. So to understand alternative pathway versus classical pathway, we need to take a step back and understand what the complement system does. So it's important to understand that antibodies don't, although that's what we're most familiar with as a part of our immune system, antibodies don't kill cells themselves. So antibodies can mark a bacteria or something like that to be gobbled up by other immune cells, but they can't directly themselves, and they don't poke the hole in the cell membrane. It's complement in conjugation or in complex with the antibody that leads to the membrane attack complex, leading to the cell damage. And it's a potent but tightly regulated system that can act in an antibody-dependent or independent manner. And those are what we call the antibody-dependent manner is the classical pathway, and the independent manner would be your alternative pathway. So classical is 3 pathways: classical, antibody-triggered, lectin. I'll lump that in with the classical system because it is also a way to get pattern recognition against sugars on a bacterial surface and lead to membrane-directed complement activation on the bacteria and killing it. And then there's the alternative pathway, which Lockman figured out or proposed in the 1970s, and then through work by Michael Pangburn and S.K. Law, they found out basically the biologic or the biochemical underpinnings of how that alternative pathway can be hypothesized to initiate complement activation. And then complement does more than just poke a hole in cell membranes. The other purpose in the fluid phase, so we can think about poking a hole in your cell membranes, but we can also think about just putting C3 on something that we don't want in our blood. So that's called opsonization. So if complement coats the surface of a bacteria or it coats the surface of an immune complex, it's going to be filtered out in our spleen and it's going to be removed. So that's, in addition to poking holes in things, the other thing that complement does is it helps filter out things that we don't want in our blood. And the other thing it does is it primes our adaptive immune system. It releases anaphylatoxins that are going to say, hey, danger signal, something is going on here. And those are just kind of split products that are churned out. So let's think about complement. But to think about complement, it doesn't occur. You need to think about whether it's occurring in the fluid phase or at the cell membrane. Bacteria don't have membrane regulators of complement, but we don't have just one membrane regulator of complement. We have many redundant membrane regulators of complement. They're factor H, which was really a fluid phase regulator, and a membrane phase regulator. They're 46, 55, CR1, and 59. CR1 is not present on endothelial cells, so that's why I put that there as a hypothesis, but we've got many backup regulators. Factor H, 46, and 55 are going to control amplification steps in the complement system. So they're on our cells to say, hey, I see an initial C3 or a C4 molecule. Before that can activate, I need to inactivate it. So that's what they do. Same for CR1. CD59 just prevents the holes in the membrane. So it kicks out the membrane attack complex before it can damage our cells. So bacteria don't have these, and that's a good thing, right? But we don't want complement that's occurring in the fluid phase to land on our cells. So we have a lot of guardians on our cells to prevent damage. So let's talk about the alternative pathway, and I'm going to focus. Sorry, I'm actually going to mention some biochemical terms on the next slide. But C3 is a big globular protein, and it's the hydrolase. So water can react with C3 and kick off the pathway. All right, so that's what actually it was Pangburn discovered, and along with S.K. Law, that found out this mechanism. That is, C3H2O can kick off the pathway. When C3H2O kicks off the pathway, so here it is. So C3 is this big globular protein. At the center, it's very well protected. So very slowly, water can reach into that site and it can activate. It can make C3 like an active C3b or C3H2O. The catch is that this is all in the fluid phase. And once you react with this thioester site, that's the site where C3 latches onto the membrane, right? So that is a dud molecule. That molecule of C3 is never going to hit the cell membrane, but in the fluid phase, it can make a convertase, but it can also be inactivated. So it's a competition between the activators and the inactivators in the fluid phase to initiate the alternative pathway. So here is BB, which would be your kind of next thing that can combine with that C3H2O. It's in the fluid phase. It can cleave. So what happens is this convertase, that would be called your first alternative pathway convertase, can cleave C3 and it can activate this thioester. Once you cleave C3 and activate the thioester, this thing is a hot It's like a grenade, but it's going to go off in like 60 microseconds actually. So if that little molecule, that thioester, hits a water molecule, it's a dud. It's going to stay in the fluid phase. It's never going to hit a cell surface. If that little hot little grenade there is along the cell surface and you're within 60 nanometers of a cell surface, you might be able to get that C3 molecule on the cell surface, and then you can go on to form a membrane attack complex on the cell surface. And that's really the damage that we care about in atypical HUS. In big studies on erythrocytes, if we throw in a strong stimulus, which I'll talk about some of them, cobra venom factor or zymacin, only 5% of the C3 we make in the fluid phase ends up hitting our cells and landing on them. And then on top of that, you then have to get past all those regulators to form the hole in your cells. So how often does C3H2O lead to activation of the alternative pathway? Given what I just said, that is C3b, it just lasts for 60 microseconds, and it has to be within 60 nanometers of the cell surface to actually have a chance of being end up on the cell. And then on top of that, we have a lot of regulators that are very good at doing their jobs. I'm a big fan of Michael K. Pangburn that did a lot of these original investigations, and he wrote a good review on this. And that was 2023. And the historical perspective on tickover, and he's one of the biggest proponents of proposing how this actually got kicked off. But he will tell you that the fundamental problem with tickover is the extremely low probability of depositing a fluid from the fluid phase, a C3b molecule, onto the surface of the cell. He says himself, the evidence for the alternative pathway activation is indirect. I won't go through those, but we do have some indirect evidence. And really, over 50 years, people have proposed, tickover. And what you really find is that no one has ever shown that it doesn't exist. And I do believe in tickover. I do think you can ultimately land something on the cell and lead to a membrane attack complex. I'm just saying it's a very weak way to activate complement. The alternative, of course, just because we haven't found definitive evidence of tickover, is that we could have just not been lucky enough or clever enough to find a better alternative. So that's how C3 is activated in the alternative pathway. But as opposed to saying alternative pathway disease or alternative pathway disorder, I would prefer, again, to say that it is an amplification loop disorder, because this is really, when you get depletion of C3 in the fluid phase, it's from the amplification loop. It's not from faster hydrolysis of the thioester. We have no way to speed up the first order kinetics of the hydrolysis at the center of the C3 molecule. That's a fixed rate. But the amplification loop is not a fixed rate. If you have a mutation in factor H or I, you can spin out. You can definitely amplify and you can deplete C3. But remember, that depletion is going to be in the fluid phase, because on the surface of our cells, we still have all these protectors. So you can churn a lot, and occasionally, if you're lucky, you might be able to form a membrane attack complex and hit your cells. The lectin and classical pathway have targeting molecules, right? They have IgG, they have IgM, and they have a lectin, right? Which is going to be targeted to the cell surface. So by definition, although I write this way up here, oftentimes, unless they're forming on an immune complex in the fluid phase, they're actually going to be able to form on the cell surface. So by definition, they're targeted, right? So they're for sure likely to end up causing more cell membrane attack complex, and cell membrane damage. And this is a description of that. So we have two different types, we have many different types of immunoglobulins, but the main ones we think about activating complement are IgG and IgM. I'm going to bring this up now, and it'll have some relevance later, but it takes 5 molecules of IgG combined with an antigen on the cell surface and C1q to do the same thing that 1 molecule of IgM does. IgM, we're born with a big repertoire of it, so everybody, even if you've never been exposed to anything in your life, you're born with some IgM that can lead to complement activation and provide your initial immunity when you're a little kid. And so that's the only other thing I would say immediately. And then IgG can be refined, but you do also have some polyclonal IgG at birth as well. So, but the same thing happens though. You can still, if you have a or lectin pathway kicking off complement, it still goes through the amplification loop. So this would still play a role. And it can still form the membrane attack complex. And I won't go into it, but you can actually get to membrane attack complex without going through the additional molecule of C3 for these convertases. We have drugs that are helpful not only to treat patients, but also to study complement. And the reason I bring these up is because I'm going to show you some basic lab data later to point this out. But C1s will inhibit just the classical pathway, and C1-INH can inhibit classical and lectin. We have a lot of inhibitors that will just inhibit the amplification loop. So these are things you may have heard of for other disorders. They're factor B inhibitors, factor D inhibitors, pegcetacoplan, iptacopan, danicopan, things like that. And then we have inhibitors that work at the cell membrane surface. That's eculizumab, a C5 inhibitor, or ravulizumab, same thing. It's going to act at the level of the membrane to prevent the holes in your cells, right? It's not going to prevent what's going on up here. It's not going to prevent amplification. It's only going to protect the surface of your cells. So complement really should be thought of, hopefully, in discussing that in detail, in two distinct phases. That is the fluid phase and the cell membrane phase. Fluid phase can occur because you have an immune complex, or because you get this tick over theory leading to hydrolysis of thioester. That's very slow. You can amplify in the fluid phase, and actually, if you amplify in the fluid phase, you very commonly form a lot of soluble C5b9. That's C5b9, that's membrane attack complex, is the other name for that never saw the surface of a cell. And that's what, when we send soluble C5b9 in a panel, that's what we're checking for. We're checking for fluid phase activation. But really what we care about is what's hitting the cell down here. And membrane-bound convertases, actually, you don't even have to deplete, and we know this even based upon other studies, you don't have to deplete complement. You don't have to have low C3 levels to have a thrombotic microangiopathy, and many other investigators have shown this as well. It's what's occurring at the membrane that's leading to the damage, and that's really what we care about in detecting. But it rarely and doesn't necessarily deplete complement. So fluid phase activation does not cause significant damage. As I said, only 5 to 10% of C3 generated through rapid amplification hits the cell surface. But the other good example of this is this guy. He's a cobra in Thailand, and he makes a venom called cobra venom factor. And this is basically a mutation that you might get in atypical HUS, right? So he makes cobra venom factor, and what happens if you put this cobra venom factor next to C3, it makes a stable convertase that none of our regulators can break apart. Okay, so this is uncontrolled activation of the alternative pathway. And what do you think happens if I give this to you or to a baboon or a mouse? Do you think they die? Uncontrolled alternative pathway activation. Are they going to get a thrombotic microangiopathy? No, no, not at all. In fact, it protects animals from complement activation as opposed to killing them, right? So we use this in xenotransplant. We've used this in many models. You can throw a bomb at the amplification loop of the alternative pathway, and the animals do perfectly fine. The baboons do fine. The mice fine. It protects them from complement activation. So fluid phase activation of complement probably protects our cell surfaces. When you see that dysregulation, it's probably a way that we've just made to protect our cell surfaces from complement. So how do we detect, and this is what I focus on, the cell complement activation that's hitting the cell membrane as opposed to the fluid phase? Well, for me, I like to make things glow. But you can actually buy this. This is a firefly petunia, and I kill plants when I try to grow them, right? So the nice thing about this plant is that if it's alive, it's glowing, and it's not just in the dark, right? This glows all the time. So this is an autonomously bioluminescent plant, and I'm not trying to advertise for them, but they are, and they're sold out for spring 2025 anyways. But the good thing about this is you'll know when it's dead. If you didn't water it right, it's going to stop glowing. So the same thing for the cells I'm using. So I take cells that have similar enzymes in them, and they glow all the time. So I take the cells and I put people's serum on it, and I see if the cells die or not, right? So the cells glow all the time, and I've got all these membrane regulators in place. And I take off the membrane regulators, actually, because we know those regulators do a really good job. So if I want to see the complement hitting the surface of the cell, I need to take off those regulators. And I actually take off not just one regulator, I can take off 2 regulators, I can take off 3 regulators, and I can get complement to hit the cell, and I can detect it in the patient's serum. And I can do this for more specificity. That is, that what a healthy control might not kill. To really, I can even use this cell line down here if I take off all the regulators to show, even without any regulators intact, whether a complement inhibitor is doing its job or not. So that's the principle of the assay. I take cells that glow, complement regulators removed. I put serum on them, and if they keep glowing, great, they didn't die, right? If they stop glowing, they're dead, just like the plant. And this is what it looks like, as opposed to being able to, like, sometimes we can, so we use an instrument to show us how well the cells are glowing. But if you take cells with complement regulators intact and you just take a normal healthy control serum and you put it on the cells, in red is is heat inactivated serum. We can use heat inactivation. It's the perfect control for complement. It shows you what would be happening to the cells if complement were not around. And in blue is the patient's serum. Here you can see there's no difference between those two lines. And I actually monitor how much the cells glow over 2.5 hours. Okay, so right here, there's no difference. Complement is not killing this cell, and this is a wild type cell. All complement regulators intact. Here, if I just take off one regulator, you see that the blue and the red still basically overlap, so I'm not killing these cells with this serum. If I take off two regulators, now I get about a 50% cell kill. That is, I'm starting to make those cells not glow. And here, if I take off three regulators, I can really even take someone that doesn't kill, cells at all, but they'll kill a cell if I take off all the complement regulators. And this is what it looks like if you just take 20 healthy controls and you run them across these cell lines. In general, as you take off more complement regulators, you get more activity. We do know that even on this 46 cell line, which is the most specific, you can get 16% of healthy controls will still kill that CD46 cell line. What happens in atypical HUS sera? Remember, most people don't kill the CD46 cell line. This is a patient that had a history, an acute sample where the patient presented with a thrombotic microangiopathy, and they have the CD46 mutation on their cells, but their serum kills, in blue, their serum kills the cells, and in red, you see what would happen if they just kept glowing. And as you would expect, eculizumab that acts at the cell membrane, it stops the death. And the alternative pathway, if I just shut off the amplification loop, it does not control the death of the cells. That is, so if I control alternative pathway amplification, I don't stop the cell death. If I put a classical pathway inhibitor, sutimlimab, that blocks very proximally right where the antibody is leading to the first formation of a convertase, it does block really just as well as eculizumab here. And remember that this is occurring in a patient where the mutation leading to complement activation was on their cells, not in their serum. So what I'm showing you here is that this is classical pathway activation, right? It's not alternative pathway application leading to this person's disease, likely, at least in an in vitro system. And at least in our cohort at Hopkins, we had about 20 different patients. We have about 7 patients with an acute thrombotic microangiopathy here. You can see that all 7 patients kill in the acute setting. And this is really in a classical pathway system. And about 50% of patients will have ongoing dysregulation or increased classical pathway activity in remission. And that's compared to TTP, another thrombotic microangiopathy, or healthy controls. These are HEC293 cells. Some people said, well, they aren't endothelial cells. And I can actually grow up endothelial cells on a plate, and I can stain them for C4. And I can show you that CM-TMA serum shows more C4 deposition compared to a healthy control. And C4 is our marker of classical pathway activation. Again, showing that this person had a classical pathway stimulus leading to their disease. And it was endothelial cells. As opposed to growing them up, it takes 2 days to grow up cells and to do this staining and show increased C4 deposition. But I can do it by flow cytometry and just take endothelial cells and stain them for C4 in the fluid phase. And again, if you have a complement-mediated thrombotic microangiopathy, you have huge increases in your C4 compared to healthy controls, but you'll notice even some healthy controls have some increase in C4 as well, which means that, okay, you might have this, but maybe you have more complement regulators on the surface of your cells to protect them compared to someone with atypical HUS or a true thrombotic microangiopathy. So, but the point is of this slide is that C4 can be detected on endothelial cells. They can be detected on a HEC293 cell. And that is, if I take sutimlimab in here and I see where the C3 came, sutimlimab is our classical pathway inhibitor. So I can put I can inhibit all of the C4 deposition. And we showed this in our paper that the classical pathway inhibition actually does a better job of controlling C3 deposition than the alternative pathway inhibitor. So, as I mentioned, we have two different immunoglobulins that primarily activate complement. We have IgM and IgG. We have enzymes that I can chop up all the IgM with, or I can chop up all the IgG with. If I inactivate all the IgM, then I should protect the cells if that's causing the complement damage. And if I inactivate all the IgG, I should, again, protect the cells if the IgG is causing the damage. And what happens is if I take the patient's serum, they will kill the cells. If I chop up all the IgG in black, I don't block the activity. Whereas if I chop up all the IgM, I can totally block the activity, showing that this is a polyreactive IgM. So it's really IgM leading to the complement activation that I am detecting here. This kind of makes sense, right? Because the first thing first of all, as I mentioned, we're all born with IgM, and you can get, atypical HUS at any stage of your life. And secondly, it's the first thing that goes up when you have an infection, when you get an influenza, when you have some sort of surgery, right? This is our first little stimulus that we get. We get a bump in IgM, and then finally we start making high affinity IgGs. So it also makes sense that infection then would be a common trigger leading to this process, leading to increased classical pathway activity that leads to the disorder. So the question is, can these cells detect alternative pathway dysregulation that we know is also a part of the disease, or really amplification loop dysregulation? And this is the patient with in what we call all pathway buffer. If I want to look at classical pathway stimulus, this is really what I've been showing you the whole time. That is, they kill the cells in all pathway buffer, and sutimlimab blocks. And this is on the CD46 knockout cell line. If I take the same CD46 knockout cell line, but I put them in a buffer that does not allow for classical pathway activity, I totally block the activity I see. That is, blue and red are the same. So that means really that the cells aren't dying if I let just alternative pathway activity occur on a CD46 knockout. So what does it take to see alternative pathway activity killing a cell? And that is, I have to totally remove all of the knockouts. So, all of the surface regulators on the surface of the cell, and only then can I take someone with a very high titer factor H autoantibody and see it actually leading to cell death, killing the cells. So again, showing the strength of the pathways. That is, classical pathway can kill a cell with regulators intact, whereas for alternative pathway to kill a cell, I have to totally remove all cell surface regulators. Showing really, again, giving another example that the damage is really coming probably more likely from the classical pathway stimulus than that amplification loop. Sorry? Sutimlimab is a complement inhibitor used for, we could go into, actually I'll finish and then I can go into that, but it's used in cold agglutinin disease, but it blocks classical pathway activity. But you can tell now I have a way. This was an acute patient, but I have a way to see a classical pathway stimulus, and I also have a way to see if you have alternative pathway dysregulation, right? So now I can take a patient's serum and I can say, do you have classical pathway activation? Do you have a alternative pathway dysregulation in the amplification loop, or do you have both? And at least in the samples that I've been able to do this with so far, I have 7 acute samples. And one of them has only classical pathway dysregulation. In the acute setting, you can expect that classical pathway, even if you don't have a mutation, drives alternative pathway dysregulation. And I do still detect that in the majority of patients, whether or not you have a variant or not. And I have one patient that had only isolated alternative pathway dysregulation. In remission, you see that of 11 samples tested, I have 4 people with purely kind of, as I said, 50% of people will have ongoing classical pathway activation. 3 patients, and especially if you have a variant, will have ongoing alternative pathway dysregulation. And 2 patients have both, 2 patients have none. But you might predict that the people, if you have a variant and you have ongoing classical pathway dysregulation, you're going to be the highest risk of relapse, okay? So there's potential in being able to look at membrane-directed complement activation to profile people and maybe predict the risk of relapse, but that's going to take prospective studies, more samples, and longitudinal follow-up. Is this the first evidence for classical pathway in an alternative pathway disease? The answer is no. Actually, the first descriptions of low complement proteins, or some of the first descriptions of low complement proteins, if you just showed a classical pathway stimulus, that is, the original description of low C3 led people to say that this was an alternative pathway disease just because they sell low C3. But that's really just showing you amplification loop problems. If you actually use a assay similar to what I use, but even in the fluid phase, they showed that you got more cell death in atypical HUS if you used something that allowed the classical pathway to occur compared to just alternative pathway. So even back in 1974, we thought that probably the classical pathway wasn't initiating stimulus. And IgM, if you read the textbooks on what you see in TMA, there's often non-specific staining for IgM in the glomeruli, less frequently C3 and IgG. But we thought it was non-specific. You know, maybe this is some evidence that the IgM is probably playing some role in the disease. And on top of that, if you just take other random skin biopsies, even if you aren't looking at the kidney, people have shown, although they didn't offer an explanation, that there's diffuse C4D staining on these skin biopsies as well. And C4D is the marker of classical pathway activation or lectin pathway activation, not alternative pathway. And we know that C4D is a common finding in thrombotic microangiopathy as well. But it's also a common finding in lupus, etc. So it's not specific at all. And these people showed that you could take cells, endothelial cells, and kill them with atypical HUS serum. And they showed that through a variety of methods that there were some IgM and IgG antibodies in atypical HUS sera that were killing cells that weren't present in TTP. So again, just more evidence. You know, we aren't the first ones to say classical pathway is playing a role here. And then this is a well-described patient with a variant in C2, a gain of function variant. And, you know, that was 2021. And for sure they had atypical HUS. And, but they also suggest that maybe we should be looking a little bit more at the classical pathway because C2 is only a component of the classical pathway. It's not a component of the alternative pathway at all. So CM-TMA, it's a disorder of the alternative pathway amplification loop, and it's probably a disorder of the classical pathway driven by IgM. For sure, germline variants lower a threshold for the disorder, but you still need your stimulus. Okay, and just like any autoimmune disorder, we don't know why some people get a stimulus leading to the autoimmune disorder and others don't. But that's probably why, you know, the daughter got the CM-TMA or the atypical HUS, but the father didn't, right? Both of them had a germline variant that lowered a threshold for cell damage, but, you know, only one of them got the disorder. And then there are all sorts of, you know, we get denuding of the glycocalyx. Some people may have higher or lower expression of membrane regulators on the surface of their cells compared to others. But that's, you know, less easily quantifiable, okay? But what I can easily quantify is whether you have a classical pathway stimulus or not. So the key takeaways are that alternative pathway is difficult to activate on a cell surface. That complement is tightly regulated, but you can amplify it very easily. IgM polyreactivity can trigger the disease in susceptible individuals, and it's a multi-hit disease, not just an alternative pathway disease. And we have new tools that can directly probe for membrane-directed classical pathway and alternative pathway dysregulation. Those are the future directions. Thank you to the patients, Dr. Brodsky, all the lab, a lot of mentors and collaborators as well that I'm working with. Thank you.
Thank you. I just a question from a patient's point of view. You said you had seven or nine patients in this testing.
Well, the acute setting, yeah, and those are the most valuable because we don't know how long that classical pathway stimulus hangs around. As I said, I can test you in remission. It doesn't tell me whether you had the stimulus, you know, at the time of your disease or not.
Does that make a difference in your testing process? The number of patients who actually, whether they're in remission, send you or come in to see you and give you their.
Yeah, no, it's tough. I mean, it's tough to recruit patients to get these samples. These are very valuable samples. People use them for other research, right? They can't be on to do this testing. This is functional testing. You need to be able to poke a hole in the cell. If you're on ravulizumab or eculizumab, you can't poke the hole in the cell. But I can do the flow cytometry, right, on the HMEC1 cells, and I can show that they have increased C4D even if they're already on the eculizumab. So it's tough because we, first line, we try to put a lot of patients, and we should, on C5 inhibitors right away, right? It's tough to get the samples where people haven't been on the inhibitors yet.
So the question would be helpful with something to the community. Like, we drove up with my daughter from Charlotte to Baltimore.
Well, yeah, there should be other easier mechanisms for you to do that. I think we'll talk to Dr. Chaudhary and USTMA, maybe like, next year a mobile blood drive or something like that. If anybody's interested in doing samples, testing, things like that. Dr. Chaudhary has mentioned that to me. She is one of the organizing people, but that would be way easier than driving up or shipping samples across country. But that is also an option, is to ship the samples. Any other questions?